How to embed pods in PeptiGels. Pods crystals are shipped sterile and dried in vials containing 50 million or 250 million pods. Always handle pods using sterile technique in a flow cabinet. Pods are typically reconstituted at 200 million pods per milliliter in PBS and vortexed. First, aliquot the desired number of pods for your experiment into a new microcentrifuge tube. Centrifuge pod suspension at 3000 G for 5 minutes. Being careful not to disturb the pod's pellet, remove and discard the supernatant. Resuspend pods in cell culture media, either with or without cells, depending on your experimental design. Select which PeptiGel you'd like to use for your experiment. Allow PeptiGel to warm up to room temperature. Using a positive displacement pipette, transfer PeptiGel to a new microcentrifuge tube. Next, add your pods and cell suspension to PeptiGel. Ensure your final mixture has a 1:4 media to gel ratio. Dispense the pods and cell suspension into the PeptiGel slowly. Mix by filling the pipette with gel from the bottom of the microcentrifuge tube. Then slowly release near the top of the mixture. Always maintain the pipette tip within the mixture to minimize introducing air bubbles. To remove bubbles after mixing, centrifuge at 2500 G for 1 minute. Dispense mixture into wells of a multi-well plate. Ensure the entire surface of the well is covered. Add enough cell culture media to each well to cover the PeptiGel. Place the well plate in the incubator at 37 degrees Celsius. Change the media 2 to 3 times within 1 hour to ensure homogeneous diffusion of media nutrients. Inspect your wells under the microscope. Pods and cells should be uniformly distributed within the PeptiGel.